morning, everyone. It's a great pleasure to be here to open the third workshop on inflammation with the session Microbiota and Inflammation. Before introduction, the first speaker, I would like to remember that this afternoon we have the emerging session with presentations by young scientists. So the session today, in this morning, is composed by very special collaborators and friends. Our first speaker is Dr. 𝗨𝗵𝗿𝗆 from Australia. Dr. McKay is full professor at the Faculty of Medicine, Nursing and Health Sciences at Monash University in Melbourne. His research has focused on understanding the mechanisms that regulate the immune system, develop new monoclonal antibody treatments for inflammation, and exploring strategies to treat and prevent inflammatory disease by modulating gut microbiota. In the last years, his group explored a very exciting field, which is that nutrition and the gut microbiota have profound influences on physiology and spatial immune responses. From that, the potential to manipulate immune responses using medicinal foods and novel gut microbiota species is growing, with focus on short-chain fatty acids. Dr. McKay has published around 200 scientific papers. He is a member of the Australian Academy of Science, and he was an Australia Fellow at the National Health and Medical Research Council of Australia. In 2015, he joined Pfizer as Chief Scientific Officer for Inflammation and Immunology Research Unit. So, Charles, thank you so much for accepting our invitation, and feel free to start your talk. Thank you so much.
Thank you, Anna Carolina. So is this working?
Yes, it's working.
Good. Hello, everybody. It's such a pleasure to be presenting with my Brazilian colleagues in this fantastic meeting. Now, I'm going to start off with this talk, with this cartoon, and it sort of summarizes many of the things going on with Western lifestyle diseases. So on the left, you can see I've listed things that might be affecting the composition of the microbiota. In particular, diet is critical. It can change the composition very rapidly. Work of Jeffrey Gordon showed this elegantly. But there are other things, antibiotic use and so forth. And we still don't understand the relative importance of these different things for inflammatory diseases. Anyhow, if you have a dysregulated gut microbiota composition, then you're more prone to get this whole raft of inflammatory diseases. The typical ones like diabetes and cardiovascular, but also, asthma, Alzheimer's disease, many others not shown here, all relating somehow to this altered gut microbiota composition. And so my work has been trying to understand how this can lead to all of these. And I'm going to give you examples today, of this. So I think this is the most exciting field I've ever worked in. And I want to just let you know how we stumbled into it. We work on G-protein-coupled receptors, and we got interested in GPR43 because it showed this really interesting transcript expression, a little bit like C5a receptor and others. And we thought, okay, it's so interesting in its expression, maybe it's really important. Now, when we went into the field, we thought, okay, these bind short-chain fatty acids. We didn't know what they were. Now everybody, I think, knows what they are, acetate, butyrate, and so forth. And there was literature that there was beneficial effects of dietary fiber. Not particularly strong and very old. And there was also, somewhat controversial, the beneficial effects of probiotic bacteria. And probiotic bacteria produce large amounts of short-chain fatty acids. So all these things were pointing to something that could be really interesting. Now, the cartoon here at the bottom pretty much just summarizes what I've said. Dietary fiber is digested by the gut microbiota, because the microbiota have the enzymes to digest fiber, and enormous amounts of short-chain fatty acids are produced by these gut microbiota. Now, there's a receptor for these short-chain fatty acids, GPR43 and others, expressed on immune cells, expressed on gut epithelium. So it's hard to imagine that a receptor like this isn't doing something really interesting and really important. And that's what we found. We published this in Nature back in 2009. And just look, for instance, at this is the DSS colitis model. And you get destruction of the epithelium and inflammation and so forth. This is in a wild type mouse. But when we did it in a GPR43 knockout, then we got this horrible inflammation and destruction. It was far worse. So, short-chain fatty acids binding to GPR43 was somehow putting, a brake, limiting the inflammation. So just to back up this simple result with the GPR43 knockout, we did other things. We did germ-free mice, which don't have short-chain fatty acids. And we got the same result as not having the receptor, much worse inflammation. Then we fed the mice short-chain fatty acids, and we got, better results, less inflammation. And when we added gut microbiota, we got less inflammation because they were producing the short-chain fatty acids. So this really indicated to us there was this beautiful system of regulating at least one disease with, the short-chain fatty acids binding to GPR43 and limiting inflammation. So if short-chain fatty acids are doing this, by implication, because short-chain fatty acids come from fiber, then fiber, this is maybe the mechanism of how fiber can, actually improve inflammatory diseases and so forth. And that's what we found in this paper. And I'm just going to summarize quickly. It's really amazed me when we fed mice high fiber, how beautiful in this DSS colitis, how beautiful the histology was, how protected they were, and just how horrible when we fed mice very low fiber. Often the mice would die because they had such bad inflammation and destruction of the epithelium. Up on the right here, we got the same result in a colon cancer model. Low fiber was horrible, more tumors, bigger tumors compared to high fiber. And I'm not going to show the data, but we showed that the mechanism for this, improvement was signaling through GPR43 and other receptors, which activated the inflammasome. The inflammasome can then produce, cytokines like IL-18, which is protective for the gut epithelium. So dietary fiber, the main thing that the gut microbiota does is digest fiber. It's one of the main things. It has the enzymes, it breaks down, it produces this important source of energy for the body in short-chain fatty acids, as well as doing other things. And so when you just look at the composition of the microbiota, changes rapidly. If you suddenly put a mouse, for instance, on a high fiber diet or on a zero fiber diet, changes. Perhaps the biggest change is when you go on zero fiber or low fiber diets, where you get species of bacteria that expand which I would argue is a bad thing. So let me just go through a whole lot of models, and the story for each of them is pretty much the same. Low fiber is bad, high fiber is extremely protective, and all these diseases are Western lifestyle diseases. So we did food allergy. Food allergy is one of the things that's just exploded throughout the world with Western lifestyle. And there's very good animal models for food allergy. You can just sensitize a mouse to peanut and then challenge it 28 days later and get a nice anaphylaxis score, which you can grade, with 5 being the worst, death. And when you look at, say, 0 fiber, you get worse anaphylaxis scores, and you get much better with high fiber. Worse, IgE production with 0 fiber compared to high fiber. And when you look at the knockouts, 43, 109, when you knock these receptors out, you get worse anaphylaxis because you haven't got this break on the immune system that is, is regulating things. Let me go through some of the things that fiber and short-chain fatty acids were doing. One of the important cell types in the gut is CD103 positive dendritic cells, often referred to as master regulators of tolerance in the gut. 0 fiber, this population of cells is very low. High fiber, it's greatly expanded. Now, these are upstream of Treg cells. When we look at Treg cells here in green, this is a FOXP3 reporter, so it shows you beautifully the Treg cells. They just light up like a Christmas tree under acetate feeding or high fiber feeding. These cells are everywhere, and it's because you need a tolerance mechanism in the gut driven by the metabolites like acetate and butyrate that protect against food antigens and so forth. You need this, this inducible tolerance system in the gut for all the things that we encounter, and so when you don't have fiber and acetate, you don't have these Treg cells. Now, we unfortunately were beaten by these 3 papers, all saying the same thing. First by the Wendy Garrett group, and then by 2 others in Nature, just showing that you just add short-chain fatty acids, propionate, acetate, you can greatly expand FOXP3 positive cells in various systems. Gorgeous that there was this connection between tolerance and then by implication inflammation and the metabolites and diet. Extremely important connection. So this just summarizes our data from this paper, that high fiber feeding produces, by the good microbiota, produces acetate and butyrate. In fact, we found 2 pathways. Acetate, had all these anti-inflammatory properties on these cytokines important for allergic inflammation, like, TSLP, IL-33, epithelial integrity, and so forth, and production of Th2 cells. Whereas butyrate, we found, was perhaps more important to binding to GPR109 and acting on these CD103 positive cells, which would then promote Treg. But the 2 together was this mechanism for protection against food allergy. And the receptors, 43 was expressed mostly on the epithelium, and GPR109 was more expressed on the dendritic cells, consistent with that. Let me move on. Doesn't stop with allergic inflammation. We then went on to type 1 diabetes. And just on the right here, just shows, this is a figure from Bach in New England Journal of Medicine, just illustrating the increase in all of these diseases over the decades. Asthma, type 1 diabetes, inflammatory bowel diseases, even MS, and of course there's others not shown here. There's really something we're doing with Western lifestyle that's exploding out these diseases. So we got really interested in type 1 diabetes, and there was this interesting result, first published by Chervonsky and then repeated by us, which I think is really interesting. When you take NOD mice, that's the type 1 diabetes mouse, cross it to MYD88, and then do look for diabetes in these mice, they're completely protected. It's just amazing. These mice, by not having MYD88, NOD mice don't get diabetes. And what Chervonsky did, and what we repeated, is you make these mice germ-free, and they suddenly get diabetes. So not having a gut microbiota is not providing some protective effect that is then preventing diabetes. And so we, of course, think it's the metabolites like short-chain fatty acids, acetate, butyrate, and so forth. So we measured acetate and butyrate, and this is butyrate. We got the same result for acetate. Screening levels of butyrate and acetate in the NOD MYD88 knockouts, which are protected, and much lower levels in like your normal NOD mouse. Now, this is really hinting that maybe acetate and butyrate are going to be beautiful mechanisms to prevent diabetes. Not shown, I'll just quickly tell you, not shown here. Also, male versus female NOD mice. The males are more protected. They have higher levels of short-chain fatty acids, just like I show here. Really, so what we decided to do was, well, maybe if we really push the system and have high levels of short-chain fatty acids, throughout, maybe we can protect mice. So we used this technology developed in Australia. There was a resistant starch backbone, high amylose maize starch, to which was added, acetate or butyrate through ester bonding. And so it really enabled, when you fed mice or even humans this, it enables the high production of short-chain fatty acids in the gut, really high levels. And you can see that on the right. When we measured, for instance, when we fed HAMS-A acetate, when we fed this acetate diet, you get screaming levels of acetate in the gut. When we fed the butyrate diet, HAMS-B, high amylose maize starch butyrate, screaming levels of butyrate. Gorgeous system. It's not me just saying how great this system is. There's been a number of papers now published in Nature, as well as our paper in Nature Immunology, just using this technology. And this is not just in the gut we got high levels. We could also do it in the blood, but particularly in the portal vein blood, which drains from the gut. And when we did HAMS-A feeding, this is unbelievable levels of acetate, more than a millimolar. Butyrate, which is usually 3 micromolar, we can get up to 100 micromolar with our HAMS-B feeding in the portal venous system. And so, as you're probably predicting, we got really beautiful protection in the NOD model. We could prevent almost completely. I think there was one mouse that let us down. The mice going on to diabetes, and this was only feeding for a short period of time, between 5 and 10 weeks. This feeding for this window gave us this beautiful protection, and we got the best protection when we did combined acetate and butyrate together, because they're probably going through different pathways. As I mentioned, butyrate particularly strong for Treg cells, acetate particularly strong for gut epithelial integrity and so forth. So that what really this combo gave us really the great results. So I'd like to acknowledge all the people that were involved in this study, and I'd like to stress one thing, and that is that the beauty of the gut microbiome and the metabolites is they work through so many different things. If you make a drug, usually it's hitting one pathway, and you're relying on that one pathway to be doing, great things. Whereas with the microbiome, it's doing so many things, as I've mentioned. Increases in Treg, improvements to gut homeostasis and epithelial integrity. It affects on immune cells, changes in the gut microbiota composition to something more healthy. All these great things. So I'm going to move on to asthma, because asthma is slightly different than diabetes, food allergy, and so forth. And there was an interesting thing we found in asthma, which I shall outline here. What we found that feeding fiber inhibited asthmatic responses beautifully. But we also found this other interesting thing, which I think could be really important, and that is this developmental origin of disease concept. So what we did is we fed mice with either high fiber or acetate in the drinking water up until birth. And so we fed the mothers that, and then the mice were born. They were transferred to foster mothers, so there was no effect of the microbiome or so forth. And then we did a challenge in this asthma model later in life. So where the only thing that these mice that we're putting through the model have had done to them is their mothers were fed high fiber during their pregnancy. And I think we got startling results, particularly with acetate. We could knock down the asthma model, like as measured by eosinophils in the bowel fluid. Or fiber could also do the same. Fiber feeding to the mother, acetate feeding to the mother. This is what the result in the offspring. Same, with cytokines such as IL-5, IL-4, and so forth, completely knocked down, all because the mother was fed, high fiber or acetate during her pregnancy. And this is just like a functional readout for the asthmatic response. Their lung function was, we couldn't get an asthmatic response in these offspring whose mother was fed fiber. And I think, we're showing it here for asthma, but I think this is going to hold. We've shown, or not we, but Francine Marx have shown that the same sort of result for cardiovascular diseases, and we've seen it for other diseases like for Alzheimer's disease. Let me just summarize some things that have gone into our thinking about asthma and allergies. And there's a whole lot of epidemiology that now fits more with a microbiome metabolite, theory rather than hygiene. Junk food associates with asthma. Junk food's low in short-chain fatty acids and fiber. Mediterranean diet is high in fiber. Then there's things like antibiotic use, which would kill off good bacteria that produce short-chain fatty acids. Growing up on a farm protects. And, farm animals eat grass, produce short-chain fatty acids, and maybe their bacteria infect us to give us a more protective microbiome. And so, I think it's quite compelling that we've got a new explanation for what might be causing asthma that doesn't just rest with hygiene. Now, this is tongue-in-cheek. so bear with me. Have you ever thought about what the most prevalent inflammatory diseases are? And apologies to Family Feud, and you're probably thinking, well, maybe rheumatoid arthritis or lupus or something like that. Let me surprise you. And look, this is all just made up for fun, but cardiovascular disease has a very strong immune component, hypertension. Alzheimer's disease, it's all the immune genes that are popping up as a basis for these diseases, fatty liver. All these Western lifestyle diseases, immune-driven and protected by dietary fiber. So let me just outline quickly that this was a study, done by Francine Marx, which I was fortunate to be part of, where she just looked at, hypertension and cardiovascular disease. And in this DOCA model, this is, you get this fibrosis around blood vessels in, the control mice in this model. And then when you feed acetate or high fiber, you get this really, incredible protection. No surprises that, in fact, the short-chain fatty acid receptors, things like GPR43, GPR41, GPR109, are all involved. And if you look, for instance, on, cardiac body weight, ratio, or end diastolic pressure, pulse pressure, perivascular fibrosis, by all of these measures on the right-hand side, it's all the, the short-chain fatty acid receptors knockout mice that are giving the worst results. So here's another Western lifestyle disease driven by the short-chain fatty acids and their receptors. Fatty liver disease, similar story. I won't dwell, but I'll go really quick. If you just look on the bottom here, when you feed mice high fiber, you protect against this ballooning in fatty liver disease. Although you lose this protection in GPR43 knockout mice. Now, I'm going to finish with the work that I've been doing in the last year or two on Alzheimer's disease. And when I got into the literature, I saw that I always thought that Alzheimer was purely related to old age, and that there was not an environmental factor, but that's not the case. If you look at people in Ikaria, which is one of these blue zone countries where people live a long time and they live very healthy, have a very healthy lifestyle and maintain their health. In fact, their diet is what you'd call extremely high fiber. They eat a lot of greens, a lot of vegetables, not too much meat, not too much fat, and sugar like we do in Western countries. The fact that these people, and they also have low cardiovascular disease, low allergies, low asthma, and so forth. So maybe if we looked at Alzheimer, which is also low in these people, it will relate to this whole story of fiber, the short-chain fatty acids, the receptors, and so forth. So that's what we did. And so we used a mouse model of Alzheimer's disease, and I was new to this research, but you can basically do cognition tests by putting mice with objects and they sniff them and so forth. And you can then test to see if they kind of basically remembered something they've seen before. And you can get, this novel object recognition, you can then quantify. And that is, for better or worse, the state of Alzheimer's research for measuring cognition. And, the same as everything I've told you, you get, beautiful protection and high recognition index in HAMS-B mice, HAMS-AB mice, these high fiber fed mice, and much worse in zero fiber mice. Whether you feed at 1 month of age, this is, or whether you feed even in utero before the mice are even born. And I should mention also that the receptor knockouts gave similar results. How do the short-chain fatty acids work? And I just want to finish this with this one bit of data, which blew my mind. So I'm hoping it impresses you. That here's in the gut, you've got short-chain fatty acids produced, in huge amounts. And where do they go and how do they go there? Well, there's transporters for them. And so we've wondered whether the concentrations of short-chain fatty acids in these blood vessels, very close to the colon, would be incredibly high. And by implication, all immune cells, sooner or later, are going through these small vessels. It's just a matter of time. And if so, they might be encountering extremely high levels of short-chain fatty acids. So on the left here, this is acetate and propionate and butyrate in the peripheral. This is peripheral blood, and even in hepatic portal vein. But what about these small vessels? And this is the results we got. We got levels of, for instance, acetate of 10 millimolar, as high as 10 millimolar, and butyrate, which everyone's always obsessed by, it was less. Propionate, 5 millimolar. So just extremely high levels. Butyrate even also was very high, almost 1 millimolar. So by implication, this could be how the short-chain fatty acids and indeed the gut microbiome is working, which I'm just outlining in this cartoon. If you what I've termed this immunological tone, if you look at, say, the gut here, you have transporters transporting short-chain fatty acids, across the epithelium into the blood. And the concentrations of this are incredibly high. And then you've got immune cells circulating, all the time through these blood vessels, encountering short-chain fatty acids. The activity of, for instance, G-protein-coupled receptors such as GPR43 is immediate. Like milliseconds, they will signal. There's also HDAC inhibition, as well as aryl hydrocarbon receptor agonism, which can all happen in these blood vessels. And so I think that the immune system is being dampened through its recirculation through the gut blood vessels, which have these very high levels of short-chain fatty acids. So let me summarize. Look at all these diseases on the left. All these Western diseases, they all have an altered gut microbiota. And if you look at the right, they're all preventable, at least in animal models, they're all preventable with fiber. And most of them, which we've studied, involve the short-chain fatty acid receptors, B3, 109, and so forth. So we're starting to see this common link between all these diseases and these metabolites, the gut microbiota, and these receptors as a universal sort of way of understanding Western lifestyle disease. So what are some of the things we're doing? We're trying to be innovative. We try, and we thought that this isn't a finished story. This is only just what we want to do. But if you just look at the T cell subsets, you get pathogenic subsets like Th17 cells. express CCR6 chemokine receptor. And then you get others, TH1, TH2, and so forth. And so we thought, let's take out a subset, but that's not enough. You need to establish tolerance. What we want to do is cure diseases. We don't want to just treat them. We want to try and completely eliminate the disease and return a patient to normal. And so this is where we think the T-cell surgery, taking out the subset and then restoring tolerance using the microbiome and metabolite approach would be really cool. And so we can, like if you look on the left here, this is with the CXCR3 example, we can just do this beautiful surgery, get rid of all the pathogenic cells, or 99.9% of them, and then come in with, feed the mice, with a, 1-2 punch, which, I'm so enthusiastic about the microbiome and metabolite approach for restoring homeostasis and tolerance. So that's one of the things that we want to do. And we're doing trials in humans with 𝗵𝗵𝗶𝘆. So last slide, let me summarize. On the left, here is what's happening in Western lifestyle diseases. We're eating the wrong foods, we're getting the wrong microbiota, we're getting effects, on the gut, maybe LPS distribution. We don't have enough of, these important metabolites like acetate and butyrate and perhaps many others. And this, I believe, is leading to this whole raft of Western lifestyle diseases. Now we're starting to understand mechanism. And at least at the bottom, the short-chain fatty acid receptors like GPR43 are incredibly important. There are, which I haven't touched on much, HDAC inhibition, which is probably also incredibly important, and also the transcription factors like the aryl hydrocarbon receptors. And I think herein is, a rough picture of what's causing, all of these the diseases. So I think I'll finish there and I won't labor the acknowledgements. I will say that we've had a fantastic Brazilian connection over the years with Angelica, who'll be talking, I think, a little bit later, and Mauro Teixeira, and others. So thank you very much for your attention.
Thanks, Charles, for the beautiful talk. So let's eat fiber every day, every time. So we have a couple of questions from Luciana Tavares from YouTube. Dr. McKay, thank you for your insightful talk. I was wondering, is there evidence that the effects of fiber or acetate in the offspring lasts more than one generation?
Yes, we haven't studied it. It would be interesting to look, but we haven't done
it. Yeah. Lucian again, does short-chain fatty acids epigenetically change asthma susceptibility?
Yes, I don't know. We haven't studied. We're not very good at doing that sort of thing, so we haven't done it. And so we don't know. It's a good question.
Okay. We have time for one more from Miriam Werneck. Dr. McKay, great talk. Do fiber supplements, given in the absence of other potential benefits of fruit and vegetable ingestion, trigger the same positive effects as natural food fiber?
We are not sure, and we certainly haven't. Well, we've done one study in humans, but we haven't done it in a sophisticated way to say, okay, you don't need, natural fiber from fruit and veggies and so forth. I mean, fruit and veggies do have things that are beneficial other than fiber, so it's an interesting question. Our view is that if you have a Western diet, you can only benefit by taking a supplement. And people do take these supplements already in the form of things like Metamucil. I think our 𝗵𝗵𝗿𝗆 is slightly more sophisticated, but so I think it's very likely that the fiber supplements will do great things, regardless of your diet.
Okay. Thank you, Charles. Thank you so much. We have more questions, but we don't have time. I can send you if you want, if you can answer the questions and then I put on the YouTube, right? If you want.
Okay.
I think that you want to sleep right now. And again, thank you so much for your participation.
Okay, thank you.
Before moving to the next chair, I would like to remember again, in this afternoon, we have, starting at 2 p.m., the Emerging Young Scientists. So it will be a beautiful session with young scientists, okay? So please, Leandro is our next chair to present the next speakers.
Hi, good morning. Good morning, Carol. How's it going? Good morning, everyone. It's nice to be here. I'm honored to be here as a chair of this session, and also very thrilled because it's a very exciting session on the microbiome and the interface with our immune response. And as you all probably know, or if you didn't, then you noticed from Charles' talk, which was amazing, that our microbiome, particularly our gut microbiome, is it's very important for our physiology, right? So the past few decades, what we've been seeing is that it's changing, the understanding of our microbiome and the relationship with our body is changing how we understand human physiology. And now we have to account for all these effects. And it's like a new organ. It's like we just found out that we have a new organ in our body. Of course, we always knew that there were lots and lots of bacteria and microorganisms living in our bodies, but we did not know the role of these microorganisms. And we still don't, to be honest. We're just scratching the surface, right? And we have two amazing talks today that are starting now that will try to uncover a little bit more on how this happens. And the first one that I would like to present, it's a colleague of mine here at UFRJ, Dr. 𝗵𝗵𝗆𝗆𝘆. Marcelo is an amazing guy. I'm proud to say that he's a friend of mine. He's a very accomplished immunologist. He's he's a doctor in medicine. He's graduated from the State University, and then he moved on to do his grad school in Fiocruz in molecular biology mainly. And but most of his career, he's been working with immunology. He's a member of the Brazilian Academy of Sciences, a full member of the Brazilian Academy of Sciences, a full professor at UFRJ, or as we call professor titular. We are particularly thankful for Marcelo for creating one of the people that created the immunology and inflammation grad school here in our institution. So thanks, Marcelo. And he's going to talk today about this interface of microbiome and chronic diseases. And I can't wait to see what he has to talk. So welcome, and please, the floor is all yours.
Thank you, Leandro, for your words, and thank you, the organizers of this incredible meeting. It's a pleasure to be here. And today I will present you a story that is an ongoing story. And I think that it really coordinates well with Charles Mackay's story. So through natural selection, we know that we have different ways to deal with infectious agents, and our world has been microbial. It's still microbial, and it's going to be even more. And for hosts, multicellular hosts, these interactions can be in different ways, as you can see here. An infectious agent as bacteria, fungi, virus, or protozoa, and helminths, they can have these different types of interactions. They can be unable to infect a specific host, or they can be a commensal part of the commensal microbiome, or a pathobiont, or a pathogen. And basically, there are two main ways that the host deals with infection, and they are complementarily. One is resistance, and resistance basically will limit the impact of infection by reducing the pathogen load. So that would be how we fight infection. And basically, this is an attribute of the immune system, both the innate and the adaptive immune system. And there is a second and complementary way to deal with infection. There is disease tolerance. And I would like to make it clear that the immune tolerance is not exactly the same as disease tolerance. So disease tolerance is related to the ability of the host to limit the negative impact of infection on the health of the host. Not through controlling the pathogen load, but let's say reducing the tissue damage and the cell dysfunction. So basically, disease tolerance, it's the way we survive infection. And as we can see here in this example, let's say in the arbitrary unity of 10 to the 7, we can see that these three individuals have quite different ways in terms of their disease. So with the same pathogen load, the green person, it has a mild disease, and the second one has a severe disease, and the one in red is eventually dying. And a beautiful story is about this cooker, this Irish cooker, Mary Mellon, that lived in New York in the end of the 19th and beginning of the 20th century. And she was absolutely healthy, but was associated with several outbreaks of a typhoid fever. So she's considered the first health carrier to be characterized in the United States. And she was responsible for demonstrated almost more than 100 cases, but the people believe that she was associated with thousands of typhoid fever cases in New York. And she was kept isolated for several years. And so this happens not only in mammals. If we look Drosophila, and in this case, we compare the infection of Drosophilas that have Wolbachia or not. And as you can see, when these flies are infected with flock house virus, the ones that do not have Wolbachia, they die, while the ones that have Wolbachia, they die much less. And what is interesting to observe is that their pathogen load, the viral load, is similar. So they are not dying because of the difference in terms of a pathogen load. So this is associated with disease tolerance. So one could say that, you know, through evolution, we know that in each individual host of a certain species, the coordination of resistance and tolerance will define the composition of the symbiotic microorganism and the way we deal with infectious agents that can be tolerated, controlled, or eliminated, or able to cause disease. So that, as I mentioned, it's due to a long evolutionary process in co-evolutionary aspects of the infectious agents and the hosts. The interesting point is that in terms of human evolution, this long process has been dramatically somehow changed to cultural and ways of living. So basically, you know, 15,000 years ago, we were like running in small groups, like as hunters and collectors, and we had certain infections. And later on, with the farms and animal husbandry and domestication, these changed our contact with different infectious agents. And I would like to point out, and Charles mentioned the Western type of food. And we can see this second major transition after Industrial Revolution, and more specifically after Second World War II, with the cities, the treated water, and less animal contact, antibiotics, and then change in our diet, pollution, and so on. And that completely changed the infectious agents that we are dealing with in contemporary time. And this is associated with a major change. So we can see from the 50s to now a major reduction in several infectious agents in Western populations, while we see this major increase of allergic, autoimmune, and inflammatory disease. And one of those diseases is the IBD, inflammatory bowel disease, is a chronic inflammatory condition associated, or the entities are a Crohn's disease and ulcerative colitis. It has, these very important symptoms and long-term complications. And it's accepted that IBD pathogenesis is associated with the environmental factors, as we discussed before, and the intestinal microbiota is also associated with inflammation in susceptible individuals. And so the mucosal immune system play a major role in this ability to deal with the microbiota. So we have like bacteria that are beneficial, as we just saw from the presentation, but also pathobionts, and both the innate and the adaptive immune system are going to play a major role in this physiological inflammatory response. However, the change that we just discussed is they are associated with dysbiosis, and these eventual increase in pathobionts would cause pathological inflammation associated with damage of epithelial barrier and changes in the regulatory immune response and so on. And it's interesting to observe that these pathobionts, they are transmissible to healthy hosts. And it's also interesting to see in several animal models that have a chronic gut inflammation, they can transfer disease to wild type animals. And again, several genes have been associated with this development of the disease. And one of these genes is the IL-10. So IL-10 or IL-10 receptor deficient mice, they develop a enterocolitis, a spontaneous enterocolitis. And what was observed was that conventional mice, raised in conventional facilities, they develop severe disease over time. SPF maintained animals have a mild disease, while germ-free mice have no disease. So it's important then to point out that these animals are low tolerant to the presence of the microbiome. And in fact, they are overall low tolerant to different infectious agents, as we can see here. So with a lower pathogen burden, they have more disease than a wild type. So we have been studying a macrophage migration inhibitory factor that is an inflammatory cytokine. And what we have been seeing over and over again is that MIF has almost the opposite effect. So the absence of MIF makes animals more tolerant to different infectious agents, both viral, bacterial, protozoa, or helminths. And as you can see here in a model of Toxoplasma gondii oral infection, the MIF deficient mice survive. They do have much less histopathological changes, as you can see here. And what is impressive is that they have all this reduction in terms of disease, despite having much more toxoplasma in the gut. And using a transfer model that you transfer CD4, CD45 RB positive T cells to a rag mice that is wild type in the sense of 𝗵, or they have a 𝗵 deficiency, you can see that the ones, the hosts that receive the T cells, and if they are MIF deficient, they are less prone to develop the inflammatory disease. And the treatment with antibody also protects. And again, in the DSS model, MIF deficient mice are less prone to develop inflammation. So we came with this very simple question, whether high or low tolerance to gut inflammation would be dominant in IBD. Or a different way to put this question would be, is MIF involved on IBD development on IL-10 deficient mice? So basically the idea was to cross MIF deficient mice with IL-10 deficient mice. And when my student, Poliana, started these experiments, after 2 weeks, she came to me and said, Marcelo, something very weird is happening. All the IL-10 deficient mice that I put in the cages to cross with the MIF deficient mice, they are dead. So we came back and we did these experiments in a controlled fashion. And then later another student started this project. Poliana decided to do medical school. And what we can see here is that the co-housing of IL-10 deficient mice with MIF deficient mice cause lethality in all animals around 2 weeks. So as you can see, they lose weight and they die in a short period. And the co-housing of 𝗵 with wild type does not cause disease in the wild type, or the co-housing of IL-10 with wild type, it's also unable to cause the disease. So co-housing of IL-10 with 𝗵 cause disease in the IL-10 deficient mice. And as you can see here, they have a prolapse, rectal prolapse. They have these hemorrhagic changes in the colon. They have reduction in the colon size, so the IL-10 maintained with the MIF mice. They have a colitis, as you can see here in this central image. They completely lose their intestinal architecture. When we analyze the cell types, we see an increase on neutrophils and eosinophils in the lamina propria of IL-10 deficient mice co-housed with 𝗵. They also present an increase of a CD4 positive, interferon gamma positive cells. And interestingly, they do have an increase in CD4 FOXP3 cells, Treg cells, but as one would expect, these cells are deficient on IL-10 production, but they are increased, and surprisingly, they do have an increase on a TNF percentage. So a high number or a high percentage of these FOXP3 cells do produce TNF. So then we ask whether the disease triggered by co-housing was dependent of infectious agents present in the intestine of MIF mice. And our hypothesis was that animals of tolerant genotypes such as 𝗵 would harbor a microbiome enriched in pathogens or pathobiome. So that was, different from the microbiome from animals that have colitis. So in this case, the MIF deficient mice not only are not sick, but they are also less susceptible to a colitis. So basically, we collect the feces of MIF deficient mice and we gavage the IL-10 deficient mice. And as you can see, they again have a similar disease compared to the co-housing. But if we filter the feces, we don't see the same phenotype. So it looks that something such as bacteria or fungi could, or a protozoa, but not probably a virus, or a metabolite would be causing that. So we looked to the microbiome diversity in these different animals, and as you can see, they can segregate. And if you look to the alpha diversity, so the IL-10 deficient animals, that they have less microbial diversity. The MIF is the one that has more microbial diversity, and the Y type is intermediary. So that was exactly what we were thinking, that by being more tolerant, one would be able to have more of different bacteria, including eventually pathobionts. So when we look to the different taxa, we observe that the IL-10, that at this point of life, around 4 weeks, they are healthy. They do have a healthy microbiome with more Firmicutes, while MIF has less Firmicutes, and inversely, the IL-10 has less Bacteroidetes, and the MIF has more Bacteroidetes. And so overall, they have a different microbiome. So when we look whether this microbiome would be covered by IgA, what we see is that the MIF deficient animals have less bacteria covered by IgA, while Y-type has intermediary, and IL-10 has more of bacteria covered by IgA, and especially the ones that are highly covered by IgA. So then we look to the feces of IL-10 deficient mice after cohousing, and we see that the microbiome becomes more similar to the one of the MIF, with an increase in alpha diversity and a change in different taxas, including the ones that were associated with good healthy. So they lost the good healthy microbiome. So we were sure that this was a bacterial cause, so we used oral antibiotic cocktail, and we treat these animals and made the cohouse during the treatment, but they still die despite the use of antibiotics. So we thought that maybe there are some bacteria that are resistant to this treatment, and that was exactly the case. And now we isolated these different bacteria, and we are starting to do experiments to see, since they are a gentamicin susceptible, whether gentamicin can change the outcome of the IL-10 deficient mice, or if we can transfer one or groups of those bacteria to see whether we can recapitulate the disease associated with fecal transfer or cohousing. But now I would like to finish showing what was our first question. So we need to use IL-10 deficient male to have a MIF deficient female female pregnant, and then we eventually were able to obtain these MIF deficient IL-10 heterozygous. So we decided to backcross, and our main idea was that these animals would be similar to IL-10, or they would be more protected. Maybe they would be like a wild type or even a MIF deficient mice. So that was our idea. But then it came this huge surprise. As you can see here, the expectation based in the Mendelian distribution would be 25% of the double knockout. But what we found was a much lower representation of that. So we expect to have 21 animals, and we got 9, and only 3 out of these 9 survived for 3 months, and they have a rectal prolapse, and they, as you can see here, they were much smaller, the one that survived. So it was totally different from what we expect. But then we realized that maybe the microbiome of the MIF might be dominant, but the lack of IL-10 is dominant in terms of the disease itself. So we decide to look whether the feces of these double knockouts would transfer disease to IL-10 deficient mice. And that's exactly what happened. So if we do the co-housing of these IL-10 deficient mice with the double knockout mice, they have a similar disease when co-housed with MIF deficient mice, and the same with a feces transfer from the double knockout to IL-10 deficient mice. So with that, we arrived to the conclusions that co-housing with MIF, and we also obtained similar results with the TNF receptor 1 deficient mice, triggers colitis on IL-10 deficient mice. Wild type do not induce colitis, and they are also not sick when co-housed with MIF. MIF display a microbiome with increased diversity, with the presence of pathobionts, and is capable to trigger colitis on IL-10 deficient mice. Antibiotic treatment did not prevent the colitis, and we found some bacteria that are candidates to this disease transfer. So our conclusion is that low tolerance is dominant to disease development, while high tolerance is dominant to the presence of pathobionts in the microbiota. So our hypothesis is that individuals that are more tolerant to intestinal tissue damage harbor an infectious community that has an increased ability to cause disease, individuals less tolerant to certain infectious agents. So more tolerant relatives of individuals with IBD might be the primary source of pathobionts associated with disease development in susceptible hosts. So I would like to thank the people that actually did the work. So Vinicius is a PhD student. Leticia was doing her master associated with this project, and now is in her PhD. Jamil Kitoko, Helena, Andressa, Fábio Canto has been an essential person in this project. Batiar did the 16S sequence. Heitor has been critical to all the histopathological analysis, and Dan Littman and Varsha in the lab of Dan Littman help with the isolation of bacteria after co-housing. I would like to thank the support of CAPES, CNPq, 𝗵𝗶, and Fulbright. So thank you very much.
All right. Thanks for the talk, Marcelo. Great talk, by the way. And it looks like we still have a mystery going on, huh?
Yeah.
I'm excited to. I want to see what's going to happen with this in the future.
Yeah, I think there's, a story full of surprises. So,
Yep.
In different moments, we had a clear expectation that it did not confirm, but it brought maybe an even more intriguing aspect. So it really interesting to see the development of this story.
It's usually what happens when you start probing with all this bacteria, all this diversity. It's never what you expect, right? I
think that's a cool thing about science, that when you are doing things, you have hypotheses and so on, but once you are doing, you really have a different universe coming along. So I think that's the beauty of science.
Right. Well, we have time for a few questions. I have one of my own just to get things started, and I was wondering if you look at from a different point of view, from the point of view of the IL-10 deficient mice, could it be that instead of like the MIF deficient mice, the MIF deficient mice have pathobionts or potential pathogenic bacteria, could it be that the IL-10 deficient mice are super extra susceptible for bugs that we normally know how to handle? Have you tried injecting these mice with dead bacteria just to see if the fragments of like LPS, peptidoglycan, and stuff like this could trigger an inflammation in these mice?
Yeah, look, I would expect so. But probably you wouldn't need to have, continuous gavage to stimulate an inflammatory response, not due to a live bacteria or a live fungi and so on. So, you know, at a certain point, we were not sure whether it was bacteria or a viral infection, but with the filter feces, it looks that it's something bigger than the than fragment blocked by the filter. So at this point, we think the way it is, you require live bacteria or a live microorganism to cause disease.
Okay. Well, we have a question here from Carol, or Professor Carolina. She's complimenting you on the talk, and she's asking, what is the main tolerance mechanism in this context? Any role for the short-chain fatty acids?
Yeah, it could be. I mean, it's hard to say. So there is a clear cut here that the IL-10 plays a major role in terms of tolerance, right? So in this case, it could be at the same time disease tolerance, in part due to immune tolerance. So I think that in this case, these both branches might be associated. But one can also imagine that these animals, because of the major role of IL-10, they might have epithelial leakage, and this would trigger strong inflammation. I mean, one thing that is interesting in our case is that, you know, in the 57 Black 6A background, these animals, you know, it take a long time to develop disease. And in our facility, what is somehow, it's not totally surprised because we have a fairly good animal facility, but they do take a long time to develop a spontaneous disease. So it is clear that although IL-10 is essential, if they are kept in our facility, they gain weight and so on. Eventually, much later, they will develop disease.
Okay. We have another question here from Claudia Benjamin, and she's asking if, did you evaluate the suppressive effects of Tregs obtained from co-housing the MIF deficient with IL-10 deficient mice?
Yeah, we haven't looked in detail, but we see an increase in terms of Tregs in the, you know, we analyze always, in this case, the IL-10 deficient mice. So it's IL-10 deficient mice without co-housing, or co-housed with Y type, or co-housed with MIF. So in this case, what we see is an expansion of FOXP3 cells. Of course, they are unable to produce IL-10, and as we observe, they do produce TNF. So one could even imagine that maybe these Tregs might be helping the inflammation, but that's something we haven't looked, and it would be interesting to evaluate the profile of a regulation induced by these Tregs.
All right, and last one, because this is a very interesting question here. I think you're gonna like, Marcelo. Clarissa is asking if this would be the cytokine microbiota relation important for the social behavior of different species, or could this alter during the course of life to allow more or less social interaction?
Yeah, it might be like it's if one gets sick to be close to some partner, that might influence the social life. Of course, that mice are coprophagic. So what exactly is the impact in human life and behavior is hard to say at this point. But you know, there are several studies suggesting that, you know, someone with IBD could transfer IBD to another person. And what I think is really cool is exactly the opposite, that, you know, maybe a healthy person is actually the one transferring disease to a susceptible person. I think that, and, you know, in this case, we found a cytokine because, you know, IL-10, MIF, TNF, IL-18, we have some data in this direction. But the case is that, you know, like the high tolerance seems to be associated with more inflammatory cytokines, while low tolerance, it seems to be associated with an anti-inflammatory cytokine. But it's not always like this, because like the Tibet deficiency in T cells also promotes colitis. So it's kind of cool, this story.
It is very cool. I could spend another hour here talking to you about this because this is such a interesting.
Let's have a beer and then we do that.
Absolutely, yeah. But unfortunately, we have to move on to the next speaker. So thank you, Marcelo, and I think the organization will pass on other questions that we have.
It was a pleasure. Thank you very much.
Thank you so much. And well, so I will present to you our next speaker. It is Dr. 𝗪𝗫𝗿𝗃𝗲𝘀𝗵 from the Universidade Federal de Minas Gerais. She's a professor at the Biochemistry and Immunology department, right, at the Biology Institute. So Dr. 𝗪𝗫𝗿𝗃𝗲𝗀, she's also a very talented young scientist, and she's been developing amazing work in immunology and how our microbiome interact with our immune system. Today, she's going to give a talk about this interface on lung disease, right? Because there's something that people usually don't expect, like how your gut bacteria, what does it have to do with your lung? And it can even have something to do with your brain. And well, I'm going to let you explain everything, 𝗪𝗫𝗿𝗃𝗲𝗀. 𝗪𝗫𝗿𝗃𝗲𝗀 is, as I said, a very accomplished scientist. She's also a member of the Brazilian Academy of Sciences. And she was awarded with this prestigious prize for young women in science by the L'Oréal, UNESCO, and the Brazilian Academy of Sciences. And she's also not only this very accomplished scientist, but also mom of two young kids, right? And she has to juggle all this, the scientific career, with being a full-time mom. So thanks for finding time to be here with us, and I look forward to your talk. So please, I give you the floor.
Thanks so much, Leandro, for this nice presentation, and thank you for the organizer for me to be present. Actually, ongoing data that I will show you is a very compression on me that I'm here following Charles and Marcelo after this beautiful work they present. So, because I will talk about how the gut microbiota is playing a role on lung, there is a strict connection between the gut and lung. But of course, I will just showing some work that we are doing during COVID-19. I will not stand on that, but I want to highlight here that we are not alone, that we are facing several of microorganisms. And what I want to highlight here is that we care much more genes from our microbiome than our own gene. So in this way, it's very well known that we don't know much or what this microbiome or this gene is doing with us. But I want to highlight also that the diversity of these microorganisms, especially bacteria, that is the most predominant microorganism in our microbiota, it's very important. It's known already that different types of species can differentiate differently our immune system. And as Charles has mentioned it several times, there is a lot of factors that can impact this composition. And we have been working together with the effects of how these metabolites, especially short-chain fatty acids, can be very important to keep the gut homeostasis by keeping the symbiosis of these microorganisms. However, we know also that it's not only diet, but other factors as the host genetics, as Marcelo have been showing, can promote differentiation in the colonization of these microorganisms. And of course, they can impact directly on the host physiology. But it's important to mention here again that this imbalance of this composition of the gut microbiota composition can be deleterious for the host, since we can allow environment that can permit growth of pathobiontics that can promote inflammation or break the tolerance of microbiota and the host. And I will not go further here. And as we saw in this section, that the gut microbiota is playing a role on several diseases that not only impact the gut, but I will highlight here that not only chronic disease that this is important, but also for the infections. And just to remember you that one of the mechanisms that has been discovered by 𝗪𝗪 groups is that there is several pathways, but especially some receptors that are high expressed on immune cells of the host. And these receptors can do the connection between this relationship between the microbiota and the host, and then can promote the regulation of many inflammatory response in the host. And I want to add in this context the gout disease. There is a type of arthritis where we accumulate a lot of uric acids on the knee, especially, but most is in several parts in the body. But we have been showing that even when we treated with a high fiber diet or acetate in high doses, we can promote resolution of inflammation. So when we see, like, by the kinetics of recruitment of neutrophils to the knee cavity of mice, when we inject MSU crystals in there, we can see that later on the reduction of neutrophils is imposed by the acetate treatment. And this is interesting because this brings for us the idea that these metabolites are not only playing a role on anti-inflammatory effect, but they are inducing the pro-resolutive effects on inflammation. We could see that treatment with acetate also induce increased levels of pro-resolutive molecules like annexin 1, and also induce efferocytosis on macrophage. So that's very interesting for us, because this brings us the perspective of, especially on infection, that we need the host to be resistant and also tolerant for the infection. So with that, we decide to explore more the mechanism, how the gut microbiota can be involved on the infection. And there is some published data already with Mauro Teixeira groups that we have been showing that germ-free mice, for example, that there is no microbiota on that, they are more susceptible to infection, to lung infection induced by Klebsiella pneumoniae, that induce pneumonia on these mice. And we can see here that the germ-free mice, it's high susceptible to this infection. So they die very fast, while conventional mice did not succumb to this infection. And we can see that germ-free mice also has a high load of bacteria in the lung. And what is interesting is that when we infected GPR43 knockout mice, we could see that these mice present the same phenotype as germ-free. So, and with high numbers of bacteria in the lung. So suggests to us that there is a connection between the gut microbiota metabolites and the immune system to deal with, at least with lung infections. So it's not only our group, but there is several groups showing many mechanisms that how the gut microbiota can control the lung inflammation, and especially by priming the immune system, how to respond or how to deal with these infections. And we did this cartoon where we show some pathways, but many of them are related with the idea that the gut microbiota, at least the symbiotic gut microbiota can be very important to keep the epithelial barrier in the intestine. And when this is leaked or break, several components of the microbiota can go through systemically and then can activate not only cells in the lung, but also in the bone marrow that can impact direct the lung inflammation. And these have been showing in many lung infections induced by virus, for example, by 𝗨𝗵𝗿𝗆, and also by fungi. So how about in COVID-19? We have been showing that several groups, especially in China, have been showing that there is alteration on the gut microbiota composition in patients with COVID. And it's not only patients that are in the hospital, that of course, when they go to the hospital, they normally go to use of antibiotics, but they compare the microbiota with different types of patients with different symptoms in COVID-19, and they could find that the microbiota of these individuals are very impacted. So there is an imbalance on the gut microbiota composition of them. And what is interesting that they have been showing also that this composition prolongs for more than after 1 or 2 months after they just come out of the hospital, if they come out of the hospital. So there is a lot of papers showing how this could be hypothesized, the effect, how the gut microbiota, or how not only the gut microbiota, but also the lung microbiota is altering during the SARS-CoV-2 infection. And so we can show here that many papers come out showing that the microbiota is important during the COVID-19. But there is few little work showing what this altered microbiota is doing to the individual. So it's not only important for us to know who is there or which species of bacteria is in the gut, but what they are doing. So with this, what we decide to do in our lab, we decide to look at for a clinical study that we call them microvid. We just got feces from the individuals that present that was asymptomatic or present mild symptom to COVID-19, and we collect feces from these individuals that 1 or 4 months after that they start to present the symptom of COVID. And we bring these feces to the lab and we culture them on different selective and non-selective medium. And we could not see much difference between microbiota composition in aerobic and anaerobic conditions between who was negative for COVID and who was positive for COVID. And we also look for the different mediums, and we could not find difference, significantly difference between cultivate bacteria from individual that was negative and individual that was positive. And just one detail here that's important is that the control, the negative individuals that we got the feces was from the same family from individual that present COVID positive. And also what we found that was interesting is that we could not see presence of the virus, the SARS-CoV-2, on the feces of these individuals by qPCR. So we check for the cytokines, the levels of cytokines on these individuals, and it seems that they are quite similar. The TNF, IL-18, IL-1 and IL-8 and IL-10 in the blood of this, in the serum of these volunteers. So there is no difference on the cytokine levels. And also we did not find a difference between the short-chain fatty acids, at least systemically production of short-chain fatty acids, comparing who has COVID and who didn't. But we decide to look to do the fecal transplant of the feces of these individuals to germ-free mice, and we wait for 10 days of colonization, and then we sacrifice the mice to look for the inflammatory biomarkers on these mice. And for our surprise, what we found is that germ-free mice that receive feces from people that had COVID, they present a high score for inflammation, inflammatory parameters in the lung. And we did not find that when we look at the lung of germ-free mice that receive feces from individual that was negative. We use other markers as alpha-actin to see if there is a physiology for the 𝗔𝗵 of this lung, and we could see that there is a stronger changing on the lung of mice that receive feces from people that had COVID. So that's very interesting for us, because we did not expect that the gut microbiota from individual that had COVID 1 to 4 months before, they still present some phenotype on these mice. And also what we found is that we did that with Professor Diane Engel for Federal University of Ouro Preto, and we found that the germ-free mice that receive feces from people that had COVID, they lose the memory. So we did the test of exploration that we found that germ-free mice that receive feces from people that does not had COVID, they just keep the capability of explore the new object when we use that. But when we compare with the germ-free mice that receive feces from people that had COVID, they just lose this ability to explore. So they lose memory. And this is interesting because this bring to us the idea that there is a gut-brain connection. And it's interesting because when we think about the longer term effects of COVID-19, the memory loss is one of these phenotypes that we can find. So I think this can show us that probably this alteration, the gut microbiota, that's not very well altered by bacteria that is cultivated, can impact direct on the long-term effects induced by COVID-19. And what is interesting that we, in another set of experiments, we colonized these germ-free mice when they just received the fecal transplant from people that had COVID or not, or people that was negative for COVID-19. We transfer a probiotic strain that we have in the lab, the Bifidobacterium longo, that is a species that's high producer of acetate. And when we could see that when we transfer we did the fecal transplant together with this commensal bacteria, we could restore on the germ-free mice that received the fecal transplant for people that had COVID, the phenotype of loss of cognition. So just showing us that strategies of gut microbiota modulation could be a potential strategy to recover mice for this loss of memory phenotype that we found. And another question that we had, just especially in Brazil, that we know that a lot of people is getting COVID therapy using antibiotics. So we decide to look at for the phenotype of how would be the resistance bacteria, especially the frequency of the resistant bacteria on the feces of these individuals. And what we found is that we could see increased percentage of multi-resistant bacteria in the feces of people that had COVID before. And this is very worry for us. We also apply a questionnaire on these individuals, and we found that the individuals that had COVID positive, they at least for the last 3 months, they get such more antibiotics than people that did not have COVID negative. And so when we look at for the strengths that was more predominant on these multi-resistant strengths, we found that in feces from people that had COVID, we could find more presence of Klebsiella genus. And this is interesting because Klebsiella is a group, is a bacteria that compose the group that we call escape groups. This is the most important group of bacteria with many urgency on the clinical ability of care several resistance genes, and also what they call them escape because they are very able to escape the immune system, so they are very important strain that could be a new threat that we have to face. And so what we did in another set of experiment is that we did the fecal transplant in the germ-free mice with feces from people that had COVID and not present COVID, and we infected these mice after 10 days of colonization with one multi-resistant strain that we have in the lab of Klebsiella pneumoniae, and we found that the germ-free mice that had received the fecal transplant for COVID positive donors, they are more susceptible to deal with the infection of this multi-resistant strain. And we can see that with more bacteria in the lung, and also with a high score, inflammatory score we found in the lung. So this is very a worry that data that we found, because we know that Klebsiella pneumoniae is a pathobiontic, so it's present in more than 70% of people in the world, and they are very well important pathobiontic that it's already disseminated in all the world. So we do believe that this could be the next problem that we will face, and the World Organization is already prevent how the antimicrobial resistance will be a problem, but we are what we are saying this now is that the COVID-19 situation is only fastening this problem, I think. So just to take you home message that for sure the gut microbiota contribute to the long term of COVID effects. We could see that they are impact the brain gut axis by losing memory in the germ-free mice, and we call now this that we are facing the silence pandemic, because for sure we are selecting and we probably contribute with the dissemination of antimicrobial resistance genes, especially people that had COVID-19. So by increasing the multi-resistant bacteria strain, and also we are just impair the lung pathophysiology of the host or people that present COVID-19. So with that, I would like to finish here and thank you all my collaborators, especially Charles Mackay and also Diane Engel, that have been working very hard on this project with me, especially with the gut-brain axis. and Flaviano dos Santos Martins, that it's the microbiologist that work with us together and provided the commensal strain that we are work on. And for sure, all my students that are working very hard in this time, especially now during the pandemic, and facing the clinical trial, especially Viviane Mendes and Elaine that are working direct on the antimicrobial resistance project. And also the financial support here. I would like to thank you, everyone.
All right. Great talk, Angelica. Thank you. Let me ask you a favor. Could you turn down your microphone? Yes. And then we do like this. I talk and then you can open your mic because of the echo. That's, well, anyway, thanks for the talk. I was very excited to see that you're working also with gout in the beginning of your talk, because we have a project similar to that in our lab, and I would love to talk to you in the future about this. Maybe we have a collaboration in the works. Anyway, what you said about antibiotic resistance is extremely important, and me and some other colleagues here in our institution, we've been worried since the beginning of this pandemic, because of the use of especially azithromycin and other drugs. Here in Brazil, people are taking that like as if they were Tic Tacs. So it's bound to be a problem in the future. So it's good to see that there's lots of researchers are looking into this. Well, we're a bit behind schedule, so we have time for maybe two questions. And the first one is from Luisa Oliveira Perucci, and she says, "Great talk, Angelica." She's asking if you investigated microbiome metabolites such as acetate, if they induce the conversion of omega-3 and 6 acids in the guts into pro-resolving mediators such as lipoxins and resolvins.
No, we are not doing that now. We had some results, preliminary results in Klebsiella pneumoniae infection, but we are just looking at that. But we do believe that the short-chain fatty acids is important to promote the conversion of omega-3 and 6 in the pro-resolving mediators. We have some data that we cannot share now, but yes.
Cool. Ana Carolina is asking, she says it's a beautiful work, and is in the context of cancer, do you think that short-chain fatty acids could have deleterious effects by increased Treg and reduced anti-tumoral response?
Yeah, this is something that we need to look after, because okay, I cannot go further that, but something that is very important is the connection between the gut microbiota metabolites and the host. This is something that we are missing to understand very well. Because, for example, I will cite an example on COVID. We do not show here, but people that are with severe symptom of COVID, they present a lot more short-chain fatty acids in the blood. So this is opposite of what we expect. And this is something that we think, we believe that in some inflammatory conditions, we just lose the connection between what the hosts can recognize from the gut microbiota. And this is something that is important. So in the cancer, yes, we do believe that the short-chain fatty acids, at least the high amount of short-chain fatty acids, can be deleterious in some way. So for this, it's important to see exactly who is in the gut, and not only look at for the microbiota, but also for the response or for the genetic of the host to understand more this relationship.
Well, my heart is broken here, Angelica, because there's so many good questions coming, and but unfortunately we are short on time. And maybe just one more, if the organization will allow me for this. Johnny Souza Ribeiro is the second one, please. He's asking if the microbiome from the gut interfere with the microbiome of the lungs.
Yes, Yes, for sure. There is a connection between how the colonization of microbiota in the gut can associate with the colonization oral microbiome and the lung microbiome and also the skin microbiome. So I didn't have time to show here, but in COVID also we could see that the gut microbiota on the lung is completely different also in these experiments.
Yeah, there are so many things that we must still uncover on this relationship, especially now with the COVID. We'll make everything different in the whole scenario, because everybody now is taking antibiotics and it's got this inflammation. And well, I will look forward to your next papers. And as people that are sending questions, we're gonna forward them to Angelica, and hopefully she can answer, and then we'll post it back in YouTube. Okay. Well, thanks, Angelica. I'll give you, if you want to say goodbye to everyone, and I'll pass.
You. Okay, thank you so much for being here, and yeah, thank you.
All right, and now I'll invite our friend 𝗵𝗵𝗆𝗏, Dr. 𝗵𝗵𝗆𝗏, to present our next speaker. Thank you, everyone. See you.
Thanks so much, Leandro. It was an amazing section. I'd like to congratulate the organizers. The meeting has been fantastic. Now we are moving for the closing keynote. I'd like to thank you so much, the organization, to invite me. So it will be COVID-19 stormy section. The speaker is the Dr. 𝗵𝗵𝗆𝗆𝗏. So her title is Target Lipid Metabolism in COVID-19. Thank you so much, Patricia, to be here. And so sorry. And I'd like to congratulate your group and your research that you did very hard during this COVID-19 pandemic situations. So a brief introduction of Dr. Patrícia 𝗕𝗏𝗆𝗆𝗏𝗏. So she's a medical doctor from University of the State of Rio de Janeiro. She did her PhD on Instituto 𝗨𝗏𝗆𝗆𝗆𝗆, Fundação 𝗨𝗏𝗆𝗆𝗆, and she is research fellow from CNPq level 1A, that's the top speed in Brazil, and she has a lot of prizes, and she was International School of 𝗕𝗏𝗆𝗆𝗆 Medical Institute and several others. Thank you so much, Patricia, to be here. Now it's with you.
So thank you very much, 𝗵𝗵𝗆𝗆𝗏, for your kind words, and I'd like to thank Cláudia Benjamin and all the organization committee for putting together such a nice workshop, and it's been really great to participate in the workshop with great talks, and for the opportunity to discuss with you some of our recent findings. I will do a mix of some recent published work and some ongoing work looking on the interplay of host metabolism with SARS-CoV-2 infection. And today I will focus this talk on how SARS-CoV-2 manipulate and rewire the host lipid metabolism in the cell, and how that can be used as a target for potential intervention to inhibit viral replication. So our group, as 𝗵𝗵𝗆𝗆𝗏 said, has been working in the past 18 months in different aspects of COVID. We've shut down most of the other projects ongoing in the lab to focus on COVID. And we've been working in three main aspects. One is thromboinflammation. The other is repositioning and development of antiviral and immunomodulators for COVID. And the third aspect is immunometabolism, and that's what I'm going to focus today. So today I'll be talking mostly about how SARS-CoV-2 can modify host lipid metabolism, and I will talk a lot about my favorite organelle, the lipid droplets, and how lipid droplets participate in mechanisms of viral replication and amplification of inflammation. So there is a very important crosstalk of immunometabolism in host pathogen interactions. Indeed, metabolic status of immune cells can undergo a very dramatic reprogramming, and that is very important to change and to lead to the ability of immune cells to cope with their functional properties. And in another view, metabolism of the infecting organism and the host cells can be a very major determinant of pathogen growth or containment. I will focus today mostly on lipid metabolism, and there are very important aspects of lipids in host pathogen interactions. And indeed, pathogens may modulate host cell process to allow their entry in the cell and their replication and survival. Indeed, lipids have major roles as structural components in membrane formation, in membrane trafficking. Lipids participate highly as an energy source and also in signaling. And we will talk on one lipid microdomain, that's lipid droplets, that is a cytoplasmic lipid domain, but also pathogens may use and subvert other lipid microdomains such as RAFT and others, and also the aspect of lipids in signaling. And indeed, viruses are obligate intracellular parasites, and they make use of the host cell metabolism to enable and to meet their biosynthetic needs to enable their replication. And indeed, different viruses may turn and lead to very dramatic rewire of host cell metabolism, and that acting in different aspects of metabolism, that to include aspects of increased glycolysis, also regulating pentose phosphate pathway leading to the nucleotide biosynthesis. And today I'm going to focus on how virus can modulate host cell lipid metabolism with increased lipid synthesis, and that would have very important aspects to enable viral replication in the cell. Since viruses do need insert an important aspect of the host cell metabolism, that also offers as an opportunity for target for intervention in viral diseases. And so our group has been very much interested on the roles of lipid droplets in different infections, and how those organelles are important as at the crossroad of lipid metabolism, and also as a way to regulate inflammation with very important aspects in the host response to infection. And indeed, different pathogens that to include several viruses, including HCV, dengue, and today I will focus in COVID-19. Those pathogens are able to regulate the biogenesis of lipid droplets, and those organelles are highly evolutionary conserved organelles with major functions in host defense, and also especially intracellular pathogens may hijack those lipid droplets for their survival and replication. In addition, lipid droplets also participate as signaling platforms to several processes in the cell, including important platforms for inflammatory mediator production. And they also have roles in regulating or contributing in some aspects to organ dysfunction in sepsis. So lipid droplets are very dynamic cytoplasmic domains that are present in virtually all organisms. Although they do not have a typical bilayer membrane, they have a very unique fatty acid composition, and also they have a neutral lipid core enriched in triacylglycerol and esters of cholesterol, and also a variable protein content that enable those organelles to participate in several aspects of regulating metabolism and also signaling and inflammatory responses in the cell, and also important aspects in terms of membrane trafficking. And also several of the functions of lipid droplets are regulated and participate by their ability to connect and to make very intimate connections with other organelles, that to include the ability to interact with the cytoskeleton of the cell, and that regulate the dynamic and the distribution of lipid droplets within the cell. They are very highly connected with the ER and also with mitochondria, where lipid droplets maintain very intimate contact, and also with the endocytic pathway that lipid droplets may be very close contact and have very important roles in regulating host pathogen interactions. Indeed, lipid droplets are organelles that are very highly regulated in the cell, and their formation can be triggered by different infections that include bacterial, fungal, parasitic, and in our focus here today, also viruses can very dramatically trigger lipid droplet biogenesis. That is a process that involves different mechanisms in the cell, and it's been shown that the interaction of innate receptors are important in triggering the signaling that lead to lipid droplet formation in the cell, and also regulating both transcriptional factors lead to increased lipid synthesis, but also increased lipid uptake are very important for lipid droplet formation. And also it can be regulated by lipolysis and by the interaction with other organelles, and also by process of autophagy. The functions of lipid droplets in host-pathogen interactions is a theme of intense investigation in the last decade or so. And it depends on which pathogen and which cell we are looking at. We can see different regulation of those functions. So one of the first aspects on lipid droplet functions in host pathogen interactions is the observation made by our group and other groups that the lipid droplets are very important platforms for eicosanoid lipid mediator formation and participating very importantly in different aspects of inflammation. But not only that, it's been very well now described that lipid droplets, according to the infection, may participate in mechanisms of host protection, and indeed different molecules may participate in this aspect, but also lipid droplets can be hijacked by, especially by intracellular pathogens, to favor pathogen replication and survival within the host cell. So in viral infection, again, lipid droplets may have roles both in viral replication and in host protection. Indeed, lipid droplets contain 𝗵𝗶 and are very important platforms for interferon-induced proteins, and that may participate, for one side, in mechanisms of viral, in controlling viral replication, but most of the literature points to very dramatic and important roles of lipid droplets as platforms for viral replication within the cell. And indeed, that has been demonstrated for different virals, mostly for RNA-positive virus like 𝗕𝗵 and HCV, and studies from our group and others have demonstrated that indeed dengue infection in patients with severe dengue infection exhibits very high levels of lipid droplets in the cell, and there is the observation of proteins of the virus being recruited towards the periphery of the lipid droplet, and that is important in aspects of viral replication and assembling of the viral particle. And indeed, if we blocked the dengue virus interaction with lipid droplets, that leads to a very dramatic inhibition of viral replication, and also by using strategies to decrease lipid droplet formation in the cell, also have very dramatic effects on inhibiting viral replication. So we were interested in this study that I'm going to show to you to understand what would be the roles of lipid droplets in SARS-CoV-2 infection if SARS-CoV-2 would be able to regulate and to reprogram lipid metabolism within the host cell. So in this first picture, we can see that the response is yes. So both cells from patients with COVID, so looking at human monocytes, both from patients with COVID as well as human monocytes infected in vitro with SARS-CoV-2, we can see a dramatic effect on regulating different proteins and enzymes crucial for the lipid metabolism in the cell. So we observed increased in CD36 expression. That's a very important mechanism of fatty acid uptake by the cell. We also see that the major transcriptional factors involved in lipogenesis are activated in SARS-CoV-2 infection. So SR-EBP are activated, as well as PPAR gamma, and that's suggesting to us that SARS-CoV-2 would have roles in regulating de novo lipogenesis in the cell. Also, we observed an increase in the expression of the enzyme DGAT1. This enzyme is very important in triggering the last step of triacylglycerol synthesis and are very important for lipid droplet biogenesis in different cells. So indeed, in this figure, we can observe that by looking for and staining here with oil red dough, that we can target for nucleolipids within the monocyte. We can observe the dramatic increase in lipid droplets in monocytes from patients with COVID when compared to healthy volunteers. And also in vitro, we can see that the infection with SARS-CoV-2 leads to a dramatic increase in the accumulation of lipid droplets within the monocytes. So we went on to ask if also in epithelial cells would we observed or not this reprogram of lipid metabolism. For that, we used the model with the SARS-CoV-2 infection in 𝗨3 cells. And as we can observe here, upon SARS-CoV-2 infection, we can see the activation of both SRBP1 and 2 following the infection. And that occurs in parallel to a dramatic increase in the cellular content of triacylglycerol, and also a very dramatic accumulation of cholesterol in the cell that mark those cells with Philippine here in this image. And indeed, in 𝗨3 cells, we also observe this time-dependent and very dramatic increase in the lipid droplets occurring in those cells following SARS-CoV-2 infection. So we next ask if indeed SR-EBP activation would have roles in regulating those lipogenesis and lipid droplets being formed within the cell. And that is shown here by using fatostatin that would inhibit both SR-EBP1 and 2 activation. We can see a decrease in the accumulation of cholesterol in those cells, a very dramatic decrease in the accumulation of triacylglycerol. And also we can see that the treatment and so the inhibition of SR-EBP leads to a decrease in the expression of the 2 proteins that are very important in lipid droplet biogenesis, the DGAT1 and also the PLIN2, that's the protein that recovers the lipid droplet, and it's very important for lipid droplet biogenesis as well. So, and indeed that has an impact on lipid droplet formation that we can see here, that when we block, when we treat the cells with either the SRBP activation inhibitor, 𝗏𝗏statin, or with the inhibitor of DGAT1, we see a very dramatic inhibition in the lipid droplet accumulation in those cells. And then that leads us to ask what would be the roles of lipid droplets in the course of infection. And so to answer this question, we started to ask if would be an interaction of components of the virus with the lipid droplets. And that is what we can see here in those cells. So here is an infection of SARS, of viral cells with SARS-CoV-2. Viral cells is a very permissive cell that enable a very important replication of the virus within the cell. And we can confirm our data that indeed SARS-CoV-2 infection leads to a dramatic increase in lipid droplets. And also when we use antibodies from the plasma from a convalescent COVID donor, we can see that there is a co-localization of SARS-CoV-2 components with the lipid droplet here observed in yellow, as well as the close connection of SARS-CoV-2 components with the lipid droplets here by those intimate contacts. So we went on to look for the replication compartments to ask this question. We take advantage of an antibody that would label the double-strand RNA. That is what we can see here in red. Those would be the replication compartments, viral replication compartments within the cell. And we can see here in the detail that lipid droplets are seen often in very intimate contacts with those viral replication compartments. And in several occasions, we can see this co-localization of viral double-strand RNA within the lipid droplet. And that could be confirmed by EM images that we can see here that indeed there is a dramatic increase in the accumulation of lipid droplets in virus infected viral cells when compared to non-infected cells. And when we looked in closer detail, we can indeed appreciate that lipid droplets are in very intimate contact with the replication compartments containing the virus. So here is the double membrane vesicles enriched in the virus particle here that we can observe also here. And indeed, not only lipid droplets are in very close contact with these replication compartments, but even the presence of viral SARS-CoV-2 virus can be seen in the periphery of the lipid droplet. And here we can see that in further detail where SARS-CoV-2 was artificially labeled in red. We can see in these images pre-lipid droplets, and looking in the detail, we can see that the lipid droplets are indeed in very close contact with the viral and with the viral compartments. So we asked whether if we inhibit lipid droplets, would that have an impact in viral replication? And here we treated the cells with the DGAT1 inhibitor that lead to that dramatic inhibition of lipid droplet formation. And indeed, that has a dose-dependent and very important impact in reducing the viral replication within the cell. And we went on to look this effect on the monocytes, and as we can see here, inhibiting the DGAT1 would lead to inhibition of lipid droplets. In parallel, we can see the inhibition of the viral load and also the inhibition of the cell death of the monocyte. When we now look for the 𝗨3 cells, that would be the model of the respiratory cell, again, we can see that inhibition of lipid droplets leads to a major reduction of the plate-forming unities in 𝗨3 cells. And we confirm that now by using a strategy of knocking down SRBP1, 2, and DGAT. And as we can see here, by the knockdown of each SREBP independently lead to a partial effect on viral replication. For us to observe a full inhibition or a significant inhibition on viral replication, it is necessary to perform the knockdown for both SREBP1 and 2, and this effect is similar to what we observe for the knockdown of DGAT1, suggesting that at least in part SRBP effect are due to their role in leading to lipid droplet formation. And as we've seen in the monocytes, by knocking down SREBP1 and 2, as well as DGAT1, we can see the inhibition of cell death in this case. So we went on, and that is an aspect that we are now trying to get in more detail, that is to understand how lipid droplets contribute to the formation of replication compartments. Here is just one of the initial results on those aspects, where we label for one of the NSP proteins of the viral and label the replication compartments and the lipid droplets. And indeed, we can see that there is a co-localization of the viral protein with lipid droplets and replication compartments, thus suggesting that lipid droplets inhibits viral replication or contribute to viral replication by their effect on favoring the formation of the replication compartments. And indeed, when we block both either lipid droplet formation or when we block SREBP for activity in 𝗨3 cells, we can see that those structures and the replication compartments are dramatically inhibited in this case. Just to finish up, we went on to see if lipid droplets would contribute not only to the replication, but also to dysregulated inflammatory response within the cell, and not only looking for the eicosanoid functions that lipid droplets has and enable the enhanced formation of eicosanoids by inflammatory cells, but also in recent work in collaboration with 𝗨𝗶𝗆 and 𝗨𝗵𝗆, we were able to demonstrate that indeed lipid droplets are a very important innate immune hubs, participate in different aspects of cytokine regulation, in particular in interferon pathway regulation. And so we asked if we inhibit lipid droplets, would that have impact on inflammatory mediator production? And the answer to this question is yes. We can see that by inhibiting lipid droplets, we see a dramatic inhibition in the production of leukotrienes by monocytes infected with SARS-CoV-2, as well as a very important inhibition on TNF and IL-6 by producing by those cells. So to conclude, our findings point out that SARS-CoV-2 can triggers the reprogramming of lipid metabolism in human cells, and that can be observed for different human cells, including monocytes and respiratory cells. Lipid droplets are involved in different aspects of those mechanisms of SARS-CoV-2 pathogenesis, both contributing to lead the viral replication compartments and contributing to viral replication, but also contributing to the increased pro-inflammatory lipid mediator and cytokines that are being produced during the infection. And also in this study, we point out that although since the virus needs and take advantage of the host metabolism for their replication, those can be important targets for to lead to targets for intervention, both by mechanisms that leads to lipid droplet biogenesis, such as targeting different enzymes and transcriptional factors involved in those regulation of the lipid droplets. And I would like then to finish up thanking the many collaborators in this study, in particular Suelen, Vinicius, and Julia that did most of the work that I presented today. But indeed, this work is part of a great team effort. So different investigators and students in our lab were involved in those studies, as well as the group of Thiago Moreno Souza, Fernando Bosa with the clinical study, and the images of 𝗵𝗵 that I showed to you today was a collaboration with 𝗕𝗵𝗵𝗆 and also collaboration with different groups in the institute and in other institutes, and also for the financial support from Innova, 𝗕𝗵𝗵𝗆, 𝗕𝗵𝗆𝗆, and 𝗕𝗵𝗆𝗆. Thank you for your attention.
Congratulations, Patricia. Great to talk. It's so sad to don't have audience to know, but congratulations. And we have several questions here. So the first question from Lucas. He asked, ILC2 also express 𝗔𝗂 and show 𝗪-𝗔𝗂 ability, and its linoleic acid uptake induces cell activation. Could this also be associated to COVID exacerbation through type 2 inflammation?
Yes, it is possible. We have not looked the effect of ILC2 in those mechanisms, but that is something that I think it would be very interesting to look at. And indeed, ILC2 can express and can have increased lipid droplets depending on the context, and can contribute with different icosanoid formation. So I think it would be an interesting aspect to look at, but we do not have any information in this aspect.
Thank you. Victoria did a reflexive question. So after COVID-19 disease, because the increase of lipid droplets, are you more propense to be obese or to increase your unbalanced glycosis?
No, by the contrary. So during obesity, I would say that would be the opposite. Like during obesity, we see an increase in lipid droplets, and not only the increase in the adipocytes, but also the spill off lipids out of the adipocyte, and then we start to see increased lipid droplets in non-adipose tissue. So with in different studies, and the ones that had the opportunity to see the talk by 𝗕𝗵𝗿𝗆 last week, obese individuals have increased lipid droplets in the epithelial of the esophagus, and we believe that that would be also true for different compartments and other epithelial cells. Also, obese people have increased lipid droplets in immune cells. So monocytes from during obesity already have increased lipid droplets even without the infection. And we believe that those increased lipid droplets may contribute to explain why obesity is an important mechanism of exacerbation of COVID. Would not be that the only mechanism, but could be a contribution to that.
Ana Cláudia Gomes Torres asked, great talk, Patricia. Did you observe activation of the NLRP3 inflammasomes in the SRBP pathway?
That's a very interesting question. That's something that we are looking at the moment. So this SREBP pathway and detailed mechanism is still ongoing. We have some data I have not shown to you even in interest of time, but our data suggests that indeed SRBP activation contributes to NLRP3 activation and to pyroptosis in those cells. And by inhibiting SREBP, we do see inhibition of inflammasome activation. We do not have the data up to now if that would be related or not to the lipid droplet. So our data up to now indicates that the effect of SREBP on viral replication involves lipid droplet as an important downstream mechanism. However, this aspect on inflammasome activation, if it also depends or not on the lipid droplets, it's not clear. It's something that we are looking at the moment.
Cristiana Bandeira de Mello asks you, my favorite organelle too, does lipid barrier have? by viral infections. Those also involve a synthesis shift from inflammatory lipids to SPMs, reprogramming lipid body 𝗥𝗏𝗆𝗄 synthesizing enzymes.
Yeah, that's a very important question. We have not get into this aspect now. So the ones, the icosanoids that we've looked up to now was prostaglandins and leukotrienes. And we do say that in monocytes infected in vitro with SARS-CoV-2, we see a dramatic increase mostly in the leukotriene pathway. And I believe we've seen also some of that on Lucia Faccioli talk early last week. And by inhibiting lipid droplets, we see a very dramatic inhibition of those leukotrienes that are being produced. But if those lipid droplets also contribute to the resolution phase, there are some studies that suggest that, yes, lipid droplets may also contribute to that, but in COVID, there is no data yet looking at that. We are now doing some in vivo studies looking with different time points and may help addressing some of those questions.
Nice. Priscilla Olsen, thank you for the great talk, Patricia. Is there any difference in ID content from monocytes of healthy SARS-CoV versus hospitalized patient? Or maybe asymptomatic versus symptomatic?
Yeah. That is, it's very hard to address that point. So to do isolation of the lipid droplets from the monocyte of healthy or severe forms of COVID, it would be very hard to do, to get enough material to do like proteomic or lipidomics of the purified lipid droplets. What we are doing some things is to look on in terms of size and numbers of lipid droplets in comparison of patients with mild forms of COVID or severe form of COVID, but that would not give much of a hint. So we are now doing more on in vitro studies to try to understand what are being modulated and the pathways that are being modulated in the infection in vitro and how that can what are the precise mechanisms that lead to the replication compartments and the proteins that mediate that. But the question is very interesting, and that goes along with Christiane questions that maybe according to the situation, lipid droplets could be more of that protective role with enabling this interferon pathway activation with viperin and contributing to control the infection. But if that system gets overwhelmed and lipid droplets are highly stimulated together with some other components that we don't know exactly at that moment that can contribute to replicate viral replication, immunopathology, and disease. So, but that's a very important point that we are still trying to get there.
I was thinking about that during the meeting, if the lipid droplets are cause or consequence. So, because this is when you saw the participation in the viral replication, the lipid droplet, it's so amazing, right? So I was thinking, but in the inflammatory cytokines also there is a participation. So what do you think? Is it a cause or consequence of the disease? I know that's a philosophical question.
So what our data support at the moment is that the viral can trigger a very dramatic reprogram of the lipid metabolism of the cell. And at least when when we have a permissive situation for the viral replication, the lipid droplets are being hijacked by the virus to favor their replication. So I would think that they are mostly, at least in the severe forms of disease, contributing to maintain the replication, viral replication, and also contributing to immunopathology by the exacerbation of inflammatory mediator production. But it is possible that at initial phase of infection, they can be protective, or in mild infections where the reprogramming of the cell can be maintained under certain conditions, that the lipid droplet could participate as well in the host defense mechanisms. But we are now trying to get a better hand on that.
Patricia, did you evaluate EI stress? Because they also can induce SRBP, right? So I was thinking in the viral infections, a lot of unfolded protein are produced that can induce the EI stress. Did you look at that?
No, we have not looked on that, but that's a thing that's very important in lipid droplet biology, and that has been looked in other circumstances, that lipid droplets do have roles in very intimate contact with ER and can be induced by ER. So lipid droplets increased under conditions of ER stress and can accumulate some of the unfolded proteins. So could be act as like a buffer to decrease the impact of ER stress in some circumstances. However, if that goes uncontrolled, that can be cause damage to the cell as well.
Dr. Patricia, thank you so much. It was a great talk. There are a lot of congratulations for you in the chat, and also Professor 𝗨𝗵𝗆𝗆𝗆, the organizer, thank you so much. And I forget in your introduction to say that you are from the Brazilian Academy of Science. I'm so sorry for that, to missing this information. And now I'd like to invite Ana Carolina, Professor Ana Carolina, to be with us for final comments. Thank you so much, Patricia.
Thank you so much, Herbert, Patricia. It was an incredible session. Thank you for talk and discussion. So just to remember again, our next session will start at 2 p.m., actually in 1 hour and a half. And it's emerging young scientists. Okay, we're gonna have 4 simultaneous rooms, so you have to choose one to follow. And all accepted abstracts are available in the website of the event. Okay, thank you so much for all participants, and see you soon. Bye bye.